started this introduction to the nervous system last time, and really the emphasis there was to give you enough background in the nervous system anatomy so that you could think about the application of action potentials and signals moving through the brain and spinal cord. So we're going to do a little more detail this time through as we talk about the nervous system. We're going to do a semblance of a kind of a dissection here of the nervous system. We're going to work on the brain anatomy, but this time we're going to talk about more of the details. We're going to work on the spinal cord anatomy. We're going to talk about more of the details of the structures here. For example, you already know that the central nervous system is the brain and the spinal cord, and you already know that the peripheral nervous system is the cranial nerves and the spinal nerves. Okay, but you don't know yet about the details of the cortical areas of the brain. You don't know yet about the ventricular system, the meninges around the brain, etc. You don't know yet about the names of the cranial nerves and their functions as they leave the brain. So, like I said, before we went through this before, I just want to give you a background kind of thing so that you could move forward. Now, we also pointed out, by way of reminder, we also pointed out in the structure of the nervous system, we think central and peripheral. But when we think about functional things, let's not forget that we're talking about somatic and autonomic information, sensory and motor. And so you'll want to go back through this again, just to review so that you have background for what we're talking about in the functions of the cortex and the functions of the nerves. You can think somatic and autonomic functionally. You'll also want to review the types of neurons and their locations. As we move forward here, I'm going to show you the specifics of the presence of these unipolar neurons and talk about reflex arcs a little bit more. We're going to talk about in the last in the last presentation of this class, we're going to talk about these bipolar neurons. So make sure you go back and review the types of neurons and the types of glial cells. So this is, whenever you learned this the first time, it wasn't intended to be learned for one test section and then forget. It was background information for what you're going to be learning about the central nervous system. Okay, so that was to say, those opening slides were to say to you, I introduced the nervous system to you. Now we're going to talk more about the details of the anatomy of the brain and spinal cord. Okay, so this begins with the covering of the brain. So this slide is titled The Meninges, and this is intended to teach you, what I'm about to teach you is the presence of fluid within ventricular chambers of your brain, where it comes from, how it gets in there, and how it gets out. And all that discussion is going to be centered around the opening volley here, which is the presence of the coverings of the brain and spinal cord. So if you took the skull off, if you took bones off the vertebral column, and you looked at the tissue there, the first thing you would encounter is the covering. The coverings of the central nervous system are what are called the meninges. These coverings are split up into three distinct layers with some nuances. So let's learn them together. So the skin and the periosteum and the bone, that's all from past experiences in life. As you get inside the skull bone, the next layer is called the dura mater. The word means tough, mother. Dura means durable, tough. Mater means mother. So the tough mother, the dura mater, the outside covering of the meninges, the thickest portion of the covering of brain and spinal cord. Now, in the brain, the dura mater itself is split up into two layers. And you can see them listed here, the periosteal and the meningeal dura. Now, that has extreme importance in the coverings of the brain. Let me illustrate it for you here in one place. In the brain, there are places where the periosteal and meningeal dura are split. The periosteal dura, throughout the inside part of the cranial vault, inside the skull, is fused to the bone. So if you took a Dremel tool, and you cut around the skull and removed the skull cap, the idea then would be ultimately to take the brain out of the cranial vault. If you did that, you would have to be very careful as you remove the cap so as to keep the fused meninge off of the skull and on the brain surface. And in order to do that, you have to actually take a scalpel and slice between the meninges and the bone. You've got to slip it in here between the meninges and the bone and pull the meninges off the bone down onto the brain surface. If you don't, you're going to rip things off you don't want to rip. So that is a good that is a good illustration in a dissection phenomenon for how you would encounter the brain. Now, here's one reason why this is so important. Because in the places where the major lobes of your brain are separated, for example, you know already that there is a major split between the left and the right hemispheres of the brain. We call that the longitudinal fissure. And in that fissure, the meninges buries deep within the brain, and here it comes, this is going to spark your memory, buries deep within the brain, and in the frontal portion of the cranial vault, it attaches to a small bony projection of the ethmoid bone called the crista galli. Remember that? Now that meninges that slips down between there is actually split from the meninges that's fused to the brain. So it's one layer, and it gets there, and then it splits. Now the split itself forms a pocket, a hole, a sinus that runs along the longitudinal fissure. And that pocket contains blood. In fact, most of the blood that goes to your brain is drained in these pockets, in these sinuses. These pockets that are located between the major lobes of the brain itself. The pockets are created by a split in the periosteal and meningeal dura. They're split apart. And all the venous blood on its way out of the brain collects in these pockets on its way out. And you can connect them. I mean, these sinuses are all interconnected. They all work back to the back here and ultimately come through the temporal bone and go through the internal jugular. So you can see in this picture the pocket in the longitudinal fissure, the split between the periosteal and meningeal dura. It is called here the superior sagittal sinus. And you can see the area I'm talking about right here. So these are places of extreme importance when you're going to be doing brain surgery. You can't hit one of these with a knife because then you've got a major bleeder in the brain. So they're very important locations. Now, I'm going to emphasize to you the importance of this location as a place where cerebrospinal fluid is drained. So, as it turns out, we have fluid on our brains and in our brains. And I would like for you to know where it comes from, how it gets there, and how it gets out. The end of that story is here. Do you see these little arachnoid villi? These little arachnoid villi protrude into these venous sinuses, and they are an outlet for cerebrospinal fluid. Much of the drainage of the CSF in the nervous system passes into the venous sinuses between the periosteal and meningeal dura. Very important. Can't be blocking these things up. You get major brain swelling here when this happens. Okay, so the arachnoid villi. Now, before I go to the next layer, let me show you something. The periosteal and meningeal dura, as I said, are fused, but you know now, in places they're split apart. And in those places, you will find the major divisions of the brain lobes. And you will find venous blood there, the sinuses of the brain. Now, before we leave the dura mater, it should be pointed out that in the spinal cord, this arrangement is different. So this picture shows you the dura mater of the spinal cord. And you can see how this dura actually covers the spinal cord in much the same way that we saw in the brain, with one major anatomical difference that you should see here. The dura mater here does not have a periosteal layer. It is not fused to the bone. Instead, there's a gap between the dura and the bone. And that gap is a very important clinical gap. It is called the epidural space. So some of you recognize that name, the epidural space. It's filled mostly with adipose tissue, but it presents a safe place to put drugs that you would like to coat the spinal nerves so that there is no sensation in that area of the spinal cord. So this is a very, most of you have heard of the process of a mother about to give birth receiving an epidural. In that procedure, a needle, then a tube will be placed into this space above the dura. And you think, well, that seems a little dangerous. And you're right, it does seem dangerous. But I don't remember ever having talked to a young mother that had some negative repercussions, spinal cord repercussions of an epidural. And the reason for that is the dura itself is a dura mater. It is a tough mother. And when you place a needle between spinous processes of vertebral bones, if you are going to penetrate through the dura, that requires effort. You have to be not paying attention in order to stick the needle through the dura. Whenever a spinal tap is done, we're going to talk more about this in the spinal cord anatomy coming. When a spinal tap is done, they go in below L2. Now you have heard this, although I didn't emphasize it. The spinal cord ends at the second lumbar bone. And you can locate that by putting your hands on top of your hips. Where your thumbs meet in your back is where the spinous process of L2 is. And as long as you stick the needle in below the spinous process of L2, you will not hit spinal cord. And so that's a different procedure called a spinal tap. When the clinician wants a sample of cerebrospinal fluid, they're going to get it out of this space, which I haven't described for you yet. But in that space, below L2, there will not be this structure. It will only be spinal roots, which you're not going to be able to stick with the needle. It'd be like trying to stick a piece of spaghetti in a pot of water with an ice pick. It just moves out of the way. When you touch it, it just moves. Same thing when you stick a needle in here. And when you stick it through, you know it. When the needle hits the dura, it stops. And you have to press intentionally, and you will hear a popping noise when it passes through the dura into the space where the CSF is. So, major anatomy difference. The dura mater is not fused to the bone in the spinal cord. Now, the next layer of meninges is called the arachnoid mater. And if you're thinking spiders, then you're doing well, because you can see that there is a gap deep to the arachnoid mater where there are spider web looking extensions down to the surface of the brain. So that's where the name comes from. The anatomist looked at that and thought it looks like a spider web. So the arachnoid mater is fused to the dura mater. It is fused to the meningeal layer of the dura. And then there is a gap between the arachnoid mater and the surface of the brain, and that gap is called the subarachnoid space. In that subarachnoid space, you will find fluid. The brain itself is floating in fluid. It is completely surrounded by cerebrospinal fluid in the subarachnoid spaces. So you can see that fluid here around the brain, And you can see that fluid here around the spinal cord in the subarachnoid space. Now, let me mention the last one, and then I'm going to talk about that fluid. The last layer of the meninges is actually fused to the outside surface of the brain and spinal cord. So all of the sulcus, sulci, and gyri, the hills and valleys, the surface of the brain and spinal cord, are followed by a thin membrane called the pia mater. The pia mater. And in a dissection, if you take a little thin dissecting tool, and you pick at the surface of the brain, you can see a thin membrane covering the surface of the brain. That is the pia mater. So the subarachnoid space is between the arachnoid mater and the pia mater, and there's fluid in that space, cerebrospinal fluid. Now, the question now is, where did that fluid come from? And how does that fluid move through the system? Okay, now, this is where I start that discussion. But I'm guessing that some of you have already washed some of this information out of your mind. And you're not thinking with me. So let me go back to it. When I taught you glial cells, I told you that one of the glial cell types are ependymal cells. And I said ependymal cells are involved in the circulation of CSF, and in the next test section, I'm going to tell you about them. So notice these ependymal cells have cilia on them. They're glial cells, and the cilia are just like cilia anywhere else. They have the ability to move. And we know that the movement of the cilia of the ependymal cells is an important piece in explaining why cerebrospinal fluid actually circulates through the system. You don't just put it in the brain and leave it sitting there. It's actually moving through the system. And so I'm going to teach you how the cerebrospinal fluid gets into the system, how it moves through the system, and how it gets out. Okay, so let's start that discussion here with my ependymal cells. This is a cartoon image of structures that are found within the brain ventricles. Brain ventricles are holes found deep within the brain. Holes filled with fluid. I'm going to teach you the names of those holes and how the things move through them. These are cavities, ventricle cavities, inside the brain mass where you find fluid. Now the fluid comes from this structure called the choroid plexus. And you can see the presence of ependymal cells here that you're not surprised by. But the question is, where does the fluid come from? And the answer is, it comes from the capillaries that are part of this structure that we call the choroid plexus. So this entire structure here, cartoon for you, is the choroid plexus. And the principal parts are the ependymal cells that move the fluid through the system and the capillaries that bring the fluid into the system. What kind of fluid is in the capillary? Blood. And blood is made up of plasma and blood cells. Plasma is mostly water. So the liquid contents of the blood within the capillaries diffuse out of those capillaries, as this gray arrow is showing you, and leave that fluid inside the brain cavities, inside the brain ventricles. Where does cerebrospinal fluid come from? This is plasma. Cerebrospinal fluid is plasma that has leaked into the nervous system through the choroid plexus capillaries. Now, there will be times where a clinician is going to want a sample of the blood and a sample of the cerebrospinal fluid. And the answer to the question, why would you need both, is actually apparent on what you have already seen. Why would you want a sample of cerebrospinal fluid in addition to a sample of blood? Because the brain and the spinal cord is bathed okay, so we're getting late in the semester. But when people stand up in class, I understand their emergencies. And I should be saying this to you because y'all are all sitting here. That is unfair to me. So the cerebrospinal fluid fills the ventricular chambers, and we want to know why it's so important to get both. And the answer should be obvious in learning the anatomy. Because all of the brain and spinal cord structures are being bathed in this fluid. So whatever is in the brain is going to be in the CSF. So when you're looking for things like meningitis and other things like that, you can find that in the CSF. It might not be in the circulatory system yet, but you'll see it in the CSF. So it serves a very important clinical function. And now you should see where it comes from. It comes out of the choroid plexus into those spaces. All right, so let's track it together. So we're going to start at the beginning and work to the end. So the first two big cavities are found in the cerebral hemispheres, and they are called the lateral ventricles. So you can see them here, these lateral ventricles. They form comma or apostrophe-shaped holes within the cerebral mass. Deep in the cerebral hemispheres, you find these lateral ventricular cavities that form these apostrophe-shaped cavities within the cerebral hemispheres on both sides. So there's two of them, one on the left and one on the right. Each of those cavities is connected by way of a small channel-like opening into a third ventricle, and that little channel-like opening is called the interventricular foramen. And it connects the fluid in the lateral ventricles to the fluid in the third ventricle. The fluid in the third ventricle then makes its way through the midbrain, through a duct called the cerebral aqueduct. The cerebral aqueduct then connects the fluid in the third ventricle to the fluid in the fourth ventricle. So there are connecting pieces for each of the four ventricles. The two lateral ventricles are the big ones at the top, the big apostrophes in the cerebral hemisphere. The third ventricle is right in the center of the brain and deep. It is in a portion of the brain called the diencephalon, which you have not learned from me yet. But when I get to it, when I get to the diencephalon, when I get there, I'm going to tell you that the diencephalon is best described anatomically as the house walls around the third ventricle. So I'm going to put the epithalamus on top of it, I'm going to put the thalamus on the side walls, and I'm going to put the hypothalamus on the floor. So the components of the diencephalon surround this cavity. So the lateral ventricles sit within the cerebral hemispheres. They're connected to the third by way of the interventricular foramen. The third sits deep in the brain, in the diencephalon. And I will use it again anatomically to describe the parts of the diencephalon. The diencephalon, third ventricle here then, connects to the fourth ventricle by way of a cerebral aqueduct. This duct passes through the midbrain. I've not taught you this yet. Some textbooks call this the mesencephalic aqueduct because diencephalon refers to the deep brain structure. Mesencephalon refers to the midbrain structure. And so that's going to pass through the midbrain. It will come up again. The cerebral aqueduct connects to the fourth ventricle, and the fourth ventricle, as you can see in these two images, sits between the brainstem and the cerebellum. So the cerebellum is the next largest mass of the brain, and you can see it here in this posterior aspect. Most of the brain is cerebrum. We say we are cerebral creatures. That's true. 83% of the brain mass is cerebrum. But we also have a cerebellum that makes up about 13% of the brain mass. It's the next largest piece of brain. And sitting deep to the cerebellum, between the cerebellum and the brainstem, so here's the brainstem right here connecting to the spinal cord, is where you find the fourth ventricle. Now you can also see in this picture that from the fourth ventricle, fluid proceeds down into the central canal of the spinal cord. There's a hole right in the middle. There's a hole right in the middle of the spinal cord, and it has cerebrospinal fluid in it. So the CSF is produced by the choroid plexus. The CSF is produced by the choroid plexus. And these structures are found in the ventricles. And you can track these things from the lateral to the third to the fourth and into the spinal cord. But there is something that should be glaring to you. When I started the description, my conversation was about cerebrospinal fluid that's in the subarachnoid space. Cerebrospinal fluid that's in the subarachnoid space. I have just described for you where the CSF comes from. And I have left the cerebrospinal fluid deep in the brain and in the center of the spinal cord. The question that should be bothering you is, how does the fluid that's produced in the chambers get out here? This is the fluid we're going to want access to. I know where it's coming from. The question is, how does it get there? And the answer is, it gets there by way of openings in the fourth ventricle. The subarachnoid spaces are connected. The subarachnoid spaces are connected to the ventricular system at the level of the fourth ventricle. Now, you don't have enough life experience to know how important that is, how important this spot is. But by the time we're done with anatomy and physiology, you will know how important this spot is, where the fourth ventricle is located, between the cerebellum and the brainstem. You've heard hints of it in this class. You've heard bits. The dens comes out of the odontoid. The odontoid comes out of the fovea dentis and penetrates the ventral portions, and that causes. And this is breathing, and so we have lots more to do here. This brainstem stuff is really important. This is a critical place in the brain. And one of the things that happens here is fluid gets out of the ventricular system into the subarachnoid spaces. So to complete the circuit, how do you get the CSF out of the system and back into circulation? If you're pouring it in all the time, you have to have a way to get it out all the time. And the answer is here, from the subarachnoid spaces, through these subarachnoid villi, into the sinuses that are located all over the gaps between the major lobes of the brain. Okay, so that is the movement of cerebrospinal fluid through the system and the ventricles. Okay, let me introduce brain structure to you. This is the outline that I'm going to use to go through the brain with you. This is a picture of the brain earlier in development, and I will not ask you to do the embryology piece here of the brain, but there's terminology that comes out of this that you must be aware of. You just simply cannot talk about brain function unless you have this vocabulary. So you have to memorize these. The telencephalon is the brain area that we refer to as the cerebrum. And the three parts we're going to talk about are cortex, white matter, and basal nuclei. The diencephalon, you've heard this today, will be the second piece, deep brain areas I'm going to talk about. And we're going to talk about the thalamus, hypothalamus, and epithalamus. And I will define their positions in relation to the third ventricle. The mesencephalon is the first piece of brainstem. And you can see the brainstem is made up of these green boxes, midbrain, pons, and medulla. So when you say brainstem, this is what you mean. Midbrain, pons, and medulla. And we'll have functional things to put on each of these. The midbrain is also known as the mesencephalon, hence the name of the duct, the mesencephalic aqueduct. In fact, that's a better name in my opinion. You call it the cerebral aqueduct, but it's in the mesencephalon. The reason some textbooks call cerebral aqueduct is because it comes out of the cerebrum, I'll grant you. But it's not there, it's in the mesencephalon. So you've heard about a little bit of this midbrain already. The substantia nigra, the beginning of the nigrostriatal dopamine pathway, begins in the midbrain. The sections that I showed you in class, the clinical sections of postmortem brain tissue of individuals who died with Parkinson's, the section was taken through midbrain of the brainstem. The metencephalon will be the pons, but the cerebellum is also related. So we'll bring up a couple of things about cerebellum here. And then the myelencephalon is the medulla, and we'll spend a little time talking about the medulla and its important functions. This is the outline that I will use to talk about the brain. So let's begin with the cerebral cortex. Now, this picture shows you what are called Brodmann's areas numerically. This comes from another textbook, and it is not your job to memorize all of Brodmann's areas. Your job is to have a general idea about the cortical areas that perform critical human physiological functions. Now, as I begin this, I like to point out to students as we begin this, it's a very popular piece of misinformation in the science fiction genre about the human brain. And it goes something like this. Whenever I perform a particular function, I use a very small piece of my brain to do so. And that is true. If you are to, today we have an imaging technique that is beyond cool, called functional MRI, where an individual can be asked to perform neurological functions, thinking, seeing, processing, speaking, moving, doing things according to instructions. And we can watch the areas of the brain that are being used in order to do those functions. That is beyond cool. Now, what you notice when you see that is only small pieces of the brain are used to do these various functions. And you're about to learn that. But there are some famous science fiction movies that I'm sure some of you have seen, where the idea is if we could just advance as a species, we would be using more of our brains. And if you could somehow access more of the brain mass, it would elevate you as a human along the Darwinian trail of human evolution. Okay, so if you want to see what that really looks like, I can show it to you. Now, in years past, I've put a little video in here to make my point, because I want students to learn that human function is distributed in the cerebral cortex in particular areas in a very organized way. It's not random, it's very organized, and you can find it where these functions are. That's why they're labeled like this in the cerebral cortex. If you want to see what it looks like when all these things are used together, or the mass of the brain is being used together, then go to Google, YouTube, and type in grand mal epileptiform seizure. Because in an epileptic seizure, the brain normally begins its stimulation in the right temporal lobe. And over a period of time, sometimes seconds, sometimes minutes, the activity of the brain will be completely encompassing. The entire brain will go active. So if you've not seen a grand mal seizure before, I had a student have one in my office about 15 years ago. It is something that you do not want to experience. If you haven't seen one, the next time you see somebody talking about we need to be using more of your brain, then you'll remember that image in your head. Yeah? You remember that. I do not want to use more of my brain. I want to use only the parts of my brain that were designed to do the functions for which they are prescribed to do. Because when you start including the rest of it, things get really ugly in human function. So, with that, let's learn some of these areas. Let's learn some of the places in the brain that have these particular functions. All right, we'll start with the front. The frontal lobe, sometimes referred to as the prefrontal cortex, this extreme frontal portion of the cerebral cortex, is responsible for what most of you guys would have written down on a paper if I asked you to tell me about the special ability of human beings. So I said, okay, let's spend a little time and talk about what makes a human uniquely human. Ultimately, those conversations come back to cognitive abilities. So it's very difficult to argue from an ethological point of view that humans are anything other than animals. However, it's also very difficult to argue that there's not something special about humans cognitively. So I took a class in vertebrate ethology many years ago, and our first project was to describe what the differences between humans and animals were. And the object of the exercise was so that this enlightened professor, who was a student of Konrad Lorenz, a Nobel Prize winner, could prove to us that human beings are nothing more than animals. So I was in my naive stage of life, and I thought people would listen to objective reasoning. So I did a lot of work on this to prove the special creation of man. And I was, well, it doesn't matter what happened to me. It was not a pleasant experience. Now, if you want to hear some more of this, you'll have to stay with me to the end of the semester. Because in the last class period of this semester, we're going to talk a little bit about the way the author of Genesis presents the differences between humans and animals. And if you don't know the Hebrew, then I promise you, you have never heard this before. Because I suspect that none of you have had any preacher types in your lives that were willing to read the Hebrew to you from Genesis chapter 1 and 2. Thank goodness you have an anatomy teacher who will. So it's coming. So this is where the conversation goes here to the prefrontal cortex. People talk about higher order thinking, cognition, personality, solving complex problems. Y'all, a frontal lobotomy, which was a horrific surgical procedure done years ago, would actually steal the humanity. These people became zombie-like creatures. Look it up. Google it up. Frontal lobotomies. Horrific procedures. Done for various reasons. Taking the humanity away from them. Their personality. Now, there's a famous story in most textbooks about Phineas Gage. His name comes up in psychology textbooks as a psychologist. I see some nodding. Psychologists like to talk about him because it's a famous myth. I have read all of the background, and the textbooks do not give you the whole story, but the story serves as an illustration. Fair enough. The guy was a wonderful dude, and he was in an accident where a rod on the railway was shot through his frontal cortex. They got the rod out, and he miraculously recovered. But on the backside of the surgery, he became a profane man. Personality change. It's such a famous myth. His brain is still on display in a little museum set up for him at Harvard University. Phineas Gage's brain sits there in a vacuum-sealed case for people to look at, as though it's some kind of relic. I want to know, what are you learning, really, looking at the man's brain? I see that something passed through his cortex. So, illustrations about the importance of the cortex. Let's keep on moving back now. As we move our way back through the frontal lobe, we get into this area of the brain here, also in the frontal cortex, an area that's called the premotor cortex. Now, each area Each functional area that does something major normally has an area close to it that will light up doing various functions, okay? And so you have to start ascribing function to this. What is this area being used for? So we know that the connectivity for this premotor cortex is primarily connected into this area of the cortex called the precentral gyrus, or the primary motor cortex. That you have heard before. What is the primary motor cortex, anatomy students? What is it for? What will I find there? What's that? The motor cortex? What do you mean by that? The little man. Yeah. The motor homunculus. That's where he is. That's right. So there's a map of the human body here where we can ascribe function to the muscles. Okay, but all of you guys know that there's certainly more sophistication to muscle contraction than just starting a signal from here and going to the muscle, right? There's more to it than that. Muscle contraction is a very sophisticated phenomenon. I ask you, for you guys in this room who are athletes and you're playing Division III athletics, if it's nothing more than sending a signal from your cortex to your quadricep, then why do you continue to repeat your activity over and over and over? Does it matter? If you're going to kick a ball or hit a baseball or throw a baseball or hit a golf ball or shoot a basketball, does it matter? I mean, if I know how to do it, why can't I just do it? Why are you doing it every day? Why? Muscle memory. Now, where is that memory? You see the question? Where is the memory of that muscle? The answer is the memory is in the premotor cortex. This is the area that associates itself with the primary motor cortex in order to do the sophisticated muscle contraction of the somatic nervous system. Think that area is important? Yeah. Okay, so now let's slip down from this premotor and primary motor cortex. Let's slip down now into this area of the temporal cortex. So we're moving now from the frontal lobe into the temporal lobe. This area right here, this temporal cortical area here, is labeled as an auditory area. It's labeled as an auditory area. Right and left. Right brain, left brain, auditory areas. Now, things get a little weird here. So there's your motor cortex. And I'm going to skip ahead here for just a minute. Oh, I took the slide out. Ooh! It'll help for you to see it. You believe me more if I show Okay. So the brain is cut sagittally now. And there's the big cerebral lobe there, cerebral hemispheres, etc. Now, this is coming used later in the presentation, but you can see here there's a big connecting band here called the corpus callosum, connecting the left and the right brain. And this is where I like to bring it up. If I asked you if you're left brain or right brain, would y'all know what I'm saying?
Yeah.
Y'all know what I'm saying? If you say you're left brain, what do you mean by that?
The right side. The right side.
If I say I'm a left brain person,
More like
science and math. And if I say I'm a right brain person, what do I mean?
English, reading, anything else but science.
Okay, so I'm an arts person.
Arts.
I'm a left brain person, I'm a math and science person, I'm a right brain person. Okay, so as we're going through this, we're going to have to address this, right? Because I'm showing you functional areas of the cortex, and you're thinking, because you were brought up learning this, that there's a left brain and a right brain. And the left brain does some things and the right brain does other things, right? Where did that information come from? We need to talk about that. And this is the place to introduce it. So this structure right here connects the left and the right brain, the corpus callosum. And when I get to it, it's coming, I'm going to talk about the split brain procedures that were done to isolate left and right brain function. But I want to bring it up today as the first piece to tell you that whenever you hear auditory cortex and the temporal cortex, when you think audition is being here in the temporal cortex, you know that there's got to be something about what I hear associated with language skills. So when I speak, I hear myself speaking, and there's got to be understanding, and there's got to be a way to say these things, yes? And that's got to be tied into the visual system as well, for language, for writing and reading, okay? So we know now, it's very unusual, that 85% of the human population has the language centers associated with the auditory cortex in the left side of your brain only. Isn't that interesting? The vast majority of the human population has its language abilities on the left side. It does not mean that all of the audition is happening on the left side, because we have an equal distribution of left ear and right ear to temporal cortex, and they cross. So these temporal cortices here on the sides of my head are helping me hear. But that language, the hearing, is tied to the way I speak. If you don't believe me, listen to the way a deaf person speaks. The articulation is different. The way they hear is related to the way they talk. What you hear is related to your language. But for language, the vast majority of people have the language centers on their left side. Now just hang on to that as your first interesting observation, because when I get to the corpus callosum, I'm going to bring that up again. I just wanted you to hear it in relation to the temporal cortex. So when we get together on Friday, I'm going to continue my journey through the cerebral cortex here.